It's really a pleasure to talk to an audience such as you. We were discussing this earlier, to see cancer survivors really gives us, not only tells us why we're doing what we do, but motivates us to do even more. So it's really a pleasure to talk to this audience. So today I think what we're going to do is something a little bit different. I want to go through one of the drugs that we're developing in Genentech. It's not an immunotherapy, although we do have a line of immunotherapies that we're carefully evaluating. This is a small molecule inhibitor, and I'm going to walk you through about almost 10 years of development that we've had all the way up to now towards the end of phase 2. And then potentially this drug can also be used in melanoma. But really I want to go through the how do we end up choosing targets and then designing clinical experiments that we want to do along the way to give you a flavor of how a company or a pharmaceutical company approaches this problem of cancer. Okay? So the drug that we have targets a pathway called AKT. It's a very important pathway that's preserved from yeast to man. It's a pathway that's important in utilizing metabolism, and it's also involved in cell survival. This pathway, the AKT, is a central node of this pathway. The role of this pathway is to integrate signals coming from the cell membrane, and then it disseminates it to a variety of effectors, about 100 effectors, ultimately leading to cell survival in times of stress, for example, and then in times of growth or in times where energy is abundant, the target, the overall pathway is involved in metabolism, to enhance metabolism. So you can see why a cancer cell would want to hijack this pathway for its own benefits. For example, to grow and proliferate, to survive, and to use energy. And really, in terms of cancer therapeutics, it really makes sense that it would be potentially important to target such a pathway. A key question that we had is that, well, because it's so important, can this pathway be targeted safely? That if we give increasing doses of an inhibitor, would this drug be ever safe? And that furthermore, how do we know that we're really targeting this pathway effectively? So those were key important early components of our development. Before we get there, it was important in order to target this pathway safely, it's important to have a very selective inhibitor. So Genentech spends a considerable amount of time. This is about, this initial project started about 10 years ago, and the initial phase summarized in this slide really took about 2 to 3 years to develop. And the real question here is that, well, when you start out with a compound, how do you target just the one protein that you're interested in? And we realized that the early generation of this compound that was generated targeted a lot of proteins that looked very similar to the one that we wanted to inhibit. But by modulating just one entity that's highlighted in yellow over time, and this is a process that involved thousands of molecules, just simplified by showing 3 here, including our now drug, by modifying this entire, the top residue highlighted in yellow, we were able to get a compound that targeted just one our protein, which hits AKT. Okay. This process was aided by evaluating the structure of the binding cleft. So by evaluating in 3-dimensional, our drug, which is pictured here, and the protein cavity highlighted here, we were able to get a molecule through structure-based drug design that targets the AKT 1, 2, and 3 isoform while sparing very similar protein called PKA. Okay, so and this process, again, took about 2 years and a lot of medicinal chemistry to get at this one compound that would bind specifically to the active site of AKT while sparing other related active sites. So then, what is can this pathway be targeted safely? Well, in order to do this, we initially tested a large panel of cancer cells that we could get a hold of. And here the tall bars mean that it takes a very high dose to hit these cancer cells. A low bar means that these cells are very responsive. So what you see is that there are a series of cells in the left that are very responsive, and some of them are melanoma cells. And then a series of cells here, not melanoma cells, that are very resistant to this drug. Next, what we did is looked at, well, what are the markers here that predict sensitivity to the drug versus resistance? And it turns out that those cells that have a high phospho AKT or high levels of AKT are uniquely sensitive. And that level occurs through either mutations that activate AKT or breaks, mutations that will accelerate the overall metabolism pathway, which will act to activate AKT, or the loss of an important break, P10, which also ends up activating the AKT pathway. This type of experiment tells us that potentially maybe we could target cancer cells selectively, specifically those cells that have activation of the pathway can be inhibited by the drug, and normal cells, for example, ones that don't have activation of the pathway, either cancer cells that are not dependent on the pathway or healthy cells that are not really dependent on the pathway, maybe they could be spared from the toxicity. So that was a theory that we had, potentially that there is a therapeutic index or a window where we could give the dose at a drug that would hit the cancer cells while sparing the normal or the cells that are not able to work. To test this theory, prior to even dosing the drug in humans, we dosed the drug in mice. And some of the experiments are shown here where in these growth curves, by giving, in red is a set of mice that over time, we engrafted tumors, and over time the tumor grows. But by giving increasing concentration of the drug, the tumor shrank across 2 different mice models. And what we wanted to do is, well, let's take out tumors at these stages and find out how much do you need to inhibit the pathway in order to get efficacy. And can we do this via conventional immunohistochemical studies that were shown earlier, so looking at the slides and cancer cells under the microscope. And we also developed a new technology, a reverse phase protein array. And the advantage of the reverse phase protein array is that it allows us to analyze 100 different proteins at once. And so we wanted to evaluate, if you block this pathway, AKT, what happens to the 100 different components of the overall pathway. So some of the data is shown here in mice. As we increase the dose, we know that this one component, phospho-S6, a very important component of the pathway, gets inhibited in a dose-dependent fashion. Also, this other protein, phospholongation factor 4G, also gets inhibited via this novel RPPA technology, but also conventional microscopy. There's very little staining when we treat with the drug for these components relative to the brown staining that you see in the absence of the drug. I'll skip this slide. In addition, what we know is that when you block a pathway, the body is very smart. It can respond by activating a different pathway that would lead to essentially the same function. So if the cancer cell is very dependent on this molecule for overall survival and it's inhibited, it's possible that some cells will just upregulate a different pathway and circumvent what we're trying to do by blocking with this one inhibitor. And we can, in fact, also with our technology, pick that up. We could pick out proteins that can get upregulated in cancer cells. And the reason why this is important is that it gives us combination approaches. But if we can see this in humans, in human patients, that specific proteins are being upregulated, then we know that maybe we should combine drug A with drug B. So by developing this technology, we wanted to implement it in our clinical trials. And that's essentially what we did. So the next step, and this is a busy slide, and I'm just going to walk you through small components of it. The next approach was to now, we had a drug a way of assessing if the drug is working in patients. And the next thing to do is to actually put the drug into patients, right? So this is a classic phase 1 design where we go from a very low dose and we only test 3 patients, and we continue to escalate the dose until we find that the drug is no longer tolerated. And once we find that it's no longer tolerated, this is called a maximum tolerated dose, or potentially a recommended phase 2 dose. So in this case, we were able to march up from 25 milligrams to essentially 600 milligrams, which was the maximum tolerated dose. And as we increase the overall dose in patients, we find that there was progressively increased toxicity until 600 milligrams, where it was generally pretty well tolerated. Anything more than 600 milligrams was not well tolerated. Well, one of the questions you may have is, well, fine, you have a dose. Is that dose effective? So one of the things that we can do is PET scans on patients. And essentially, every patient at this dose had a nice PET response. And you can see that here in a lady who had breast cancer. I should mention these trials are done in all solid tumors. Some of the patients had melanoma, but it is the initial phase 1 trials are generally done regardless of tumor types. And this one patient had lots of tumors, particularly in her liver and in her vertebrae, but in the presence of the drug, the tumors went away. And the only thing that you see is some degree of radioactivity in the bladder, which is what you expect on the PET scan. So essentially, the patient had a complete PET response. One evidence that the drug is working. Several other evidence that we look at is, one is just exposure overall. And we know that at the 600 milligram exposure, the amount of drug that we're getting in is comparable to the doses that we saw in mice that led to shrinkage of the tumor. So we know that by just calculating and determining the amount of drug exposure, how does that relate to our preclinical models? How does that relate to our mice models? And we know that we were able to achieve fairly high amounts of exposure. Another way of looking at are we at effective doses? And one may wonder, well, 600 milligrams was great. Do lower doses also work? And in order to test this, we collected blood from patients, and as we increased the dose, evaluated how much are we hitting the pathway, looking at some components of the pathway. And it turns out, as we march up from 200 milligrams to 400 milligrams, we're inhibiting the pathway anywhere from 70 to 80%. That the most of the inhibition occurs pretty very early, around 200 to 400 milligrams, making us think that maybe we don't have to give such a high dose. There's a potential of giving even lower doses to patients. And then the last component is this technology that we discussed earlier, the ability to evaluate 80 proteins or 100 proteins at once, of evaluating to what degree are we knocking down the pathway, and are there proteins that are being upregulated. And in order to do that, we did tumor biopsies before starting treatment and on treatment. And these results show just a couple of proteins where as we increase the dose, we find that in, for example, PRAS40, another important downstream element of AKT, there is about an 80% inhibition of the pathway. But not only PRAS40, many different components of the blue here connotes that the pathway is knocked down. Red means that there is a parallel pathway that got upregulated in response to knocking down our protein. So this pathway is very much knocked down in a dose-dependent fashion, but that there are some patients that have upregulation in parallel pathways, making us think that perhaps there is a combination strategy for work for these individuals. Okay, so the first experiment is a phase 1 component of it to determine what dose we should be going into patients. The next part of the thought process that we engage is in what tumor types do we test such a drug? And this really is dependent on what questions do we want to ask in patients. So the main question we wanted to ask is to interrogate tumor types where we know the pathway is active. And that's That's question number one depicted here. The second question we wanted to address is, well, what are the combinations that we should be addressing? And these combinations were really born from when you inhibit the pathway, what are some of the proteins that are upregulated? So we know when we dose with chemotherapy, AKT is upregulated. When we dose with anti-hormone therapy, for example, breast and prostate cancer, we know that AKT gets upregulated. And the last is in combination with MEK. MEK is something that we'll talk about a little bit later today, is a very important pathway in melanoma. And when MEK is inhibited, at times AKT can be inhibited as well. Okay, so these were the questions that we want to address in clinic. So then we did a whole variety of combination trials, which essentially is drug A plus drug B. And the combinations of trials are done in order to find doses of both drugs, both elements to put together. In order to do these combinations, there must be a scientific rationale. We have to go back to the laboratory and show that, well, there is really a purpose to these combinations. And that's somewhat depicted here, where we're showing what is the degree of synergy, what is the degree that the two drugs work well together relative to either drug alone. So each dot here represents, colored dots represents a different chemotherapy that we tried in combination with our drug, ipatasertib. And the lower the number, the lower the combination index, means that there is broader synergy. And if it's high, it means that there is antagonism, that the two drugs don't play and they hurt each other. And as you can see with chemotherapy, it turns out that a majority of chemotherapy, whether it's docetaxel, gemcitabine, platinum, different types of damaging agents, that there is a broad degree of synergy, not only in tissue culture, in a petri dish, but also in mouse models, where we combine when we combine the two drugs, there is greater activity relative to either of the 8 drugs alone. The rationale around that synergy is that when we block with one agent, such as chemotherapy, there is upregulation of AKT. So this entire notion that the body is smart, when you block, when you treat with chemotherapy or potentially targeted therapy, at times the cells respond by upregulating a different pathway. And the thought is that by blocking both pathways, there might be greater degree of cell death depicted here. Okay. So then with this rationale, we engaged in a variety of combinations. Combination one is with paclitaxel, another combination is with docetaxel, a third combination depicted here is another chemotherapy, FOLFOX. And again, the purpose here is to march up on the dose until you get a dose that is well tolerated in humans, and that's shown in red boxes, that the doses that we're proceeding with in phase 2s. In terms of looking at efficacy, the interesting thing we did in our trial is to, with combining drug A with drug B, what we wanted is individuals to have been treated with that drug previously and then add our drug on top. So those individuals who were previously dosed had progressed, and now if we add our drug plus the old drug, what happens? It turns out that we were able to get responses in many of these patients who were previously refractory to that drug. And these responses generally occur in patients who have the pathway activated. And we know that by sequencing their tumors and looking at a variety of mutations that can upregulate the overall pathway, such that if we look at those individuals who have a mutational activation of the pathway, they were much more likely to respond relative to those individuals who were wild type or did not have a mutation in that pathway. Okay. So we know that the drug is working as predicted. In terms of where we're going currently with this agent, there are 3 phase 2 trials, and let me walk you through what we're trying to do here. One is in gastric cancer, and the reason why we chose gastric cancer initially is because the pathway, again, is activated in about 80% of individuals with gastric cancer by a variety of different mechanisms. And in this trial, we're treating half the patients with the standard therapy for gastric cancer, which is this chemotherapy called FOLFOX. The other 50% will get our drug in combination with FOLFOX. And then we'll essentially look at what is the overall response rate, what is the overall survival in population A versus B. And this trial should read out pretty soon, and it'll give us an indication of, is this drug effective in something like gastric cancer? The other tumor type that we're engaged in is breast cancer, specifically triple-negative breast cancer. And here the trial design is very similar in that half the patients are treated with conventional paclitaxel, chemotherapy that's used in breast cancer. The other half of the patients will receive paclitaxel in combination with our drug. And again, we're looking for a benefit in terms of how long is the disease controlled prior to progressing. And we'll also look at what is the overall survival of patients across the two arms. Okay? And the third disease that we're looking into is prostate cancer. And here we're looking at multiple doses of the drug, a low dose of our drug, a medium, and the maximum tolerated dose in combination with an anti-hormone therapy, and look in a 3-arm trial. Again, with the primary endpoint will be how long is the disease controlled prior to progressing, as well as we're looking at as a secondary endpoint overall survival. So 3 trials, but what about melanoma? Well, we know melanoma also has this pathway active, right? About 50% of melanoma have BRAF mutations, but again, the body is smart. For a proportion of these individuals, about 50% that have BRAF mutation, also have activation of the AKT pathway, frequently via loss of this brake, P10. And that's shown here. The other way of activating the pathway is by blocking with a MEK inhibitor or a BRAF inhibitor. The body can upregulate AKT via this reciprocal feedback mechanism. So then the thought is, well, why not combine these 2 drugs? And we're in preclinical models in mice, it really works very well when the combination of our AKT inhibitor combined with a drug that blocks the MEK pathway works very well relative to either agent alone. So then we engaged in a combination trial where both drugs were escalated in 2 different fashions. In one experiment, both drugs were given as a pill, 1 pill a day, every day, in arm A, and then in arm B, this ipatasertib, the AKT inhibitor, was given every day. And with cobimetinib, we gave high doses every 4 days of the drug. So 2 different arms, 2 different ways of giving these agents. And essentially what we find is that a lot of patients do benefit, some don't. By looking at the genetics of the tumor, we can come up with a predictive signature of those individuals who are likely to benefit. So here, these are waterfall plots, and those individuals who are below 0 are responding to therapy. Their tumors are shrinking. Those individuals, each bar is an individual tumor assessment. Those bars that are above 0 are individuals who had growth of their tumor. And by evaluating the genetics of the tumor, we're able to figure out what are the markers of response relative to resistance. In terms of the major markers, it turns out that those individuals who have the pathway activated, BRAF mutations, for example, or P10 low, are more likely to respond than those individuals who don't. So this leads us to potential future therapies of melanoma. For example, we know that BRAF is mutated quite frequently. Well, what about those individuals that have BRAF mutations, but potentially also P10 loss? Might we be able to do this combination in, might we be able to do this trial in the future? So where are we with our AKT inhibitor? Well, it's a very selective, potent inhibitor to the 3 isoforms of AKT. The AKT pathway, we now know, can be targeted safely at doses 100 to 600 milligrams. It's not really clear if you really need such a high dose. Maybe a lower dose may work just as well, and we're testing this in our current development. The drug combines very well across several standard of care treatments. The combinations that we've selected are those that cooperate with one another. Specifically, we're looking for reciprocal feedbacks where if you block one pathway, the other pathway gets activated. And then we have multiple phase 2 trials underway right now. Okay.